Turmeric. Every grandmother in South Asia has been putting this yellow root into food and wounds and warm milk for thousands of years, and every Western-trained physician who encountered this practice for the first 2 centuries of colonial medicine dismissed it as folk superstition with no biological basis. And then, in 1995, the United States Patent and Trademark Office granted a patent to 2 researchers at the University of Mississippi Medical Center for the use of turmeric in wound healing. And the Indian government challenged the patent on the grounds that turmeric's wound healing properties had been documented in Sanskrit medical texts for over 2,000 years, and the patent was revoked in 1997 in what became one of the landmark cases in the history of biopiracy. And the revocation required the Indian government to produce documented prior art showing turmeric's medicinal use predating the patent application. And the prior art they produced included references in the Sushruta Samhita dated to approximately 600 BC, describing turmeric's application to wounds and its use in treating inflammatory conditions, with a clinical specificity that made the patent application's claim of novelty impossible to defend. And the active compound in turmeric responsible for its medicinal properties is curcumin. And since the patent revocation, there have been over 10,000 published studies on curcumin in peer-reviewed medical literature, making it one of the most extensively researched natural compounds in modern pharmacology. And those studies have confirmed anti-inflammatory effects, antioxidant activity, neuroprotective properties, and potential applications in the prevention and treatment of Alzheimer's disease, multiple forms of cancer, cardiovascular disease, and depression. And the specific mechanism by which curcumin produces its anti-inflammatory effects, the inhibition of the NF-κB signaling pathway which regulates the inflammatory response, was identified by modern molecular biology approximately 2,000 years after Indian physicians had empirically identified the clinical outcome that mechanism produces. And the question that pharmacologists who have spent careers studying curcumin consistently find most genuinely difficult to answer is not whether it works, because the clinical evidence is now overwhelming, but why it took the most sophisticated medical research apparatus in human history 2,000 years to confirm what a grandmother in Varanasi already knew. Now, fasting. Every major religious and spiritual tradition in human history has incorporated periods of deliberate food abstention into its practice. The Ramadan fast of Islam, the Yom Kippur fast of Judaism, the Lenten practices of Christianity, the Ekadashi fasts of Hinduism, the periodic fasting of Buddhist monasticism, and the consistent dismissal of these practices by modern medicine for most of the 20th century was that they had no biological benefit and were purely spiritual or cultural practices with no physiological rationale. And then, in 2016, the Nobel Prize in Physiology or Medicine was awarded to the Japanese cell biologist Yoshinori Ohsumi for his discoveries of the mechanisms of autophagy, the process by which cells disassemble and recycle their own damaged components. And what Ohsumi's work revealed, and what the subsequent decade of autophagy research has confirmed, is that the primary trigger for autophagy in human cells is the absence of food, meaning that the periods of deliberate food abstention that every major spiritual tradition had been prescribing for thousands of years were triggering a cellular cleaning and renewal process whose existence modern biology did not confirm until 2016. And the specific health benefits that ancient traditions attributed to fasting, longevity, mental clarity, disease prevention, the purification of the body, map with remarkable precision onto the documented biological effects of autophagy, which include the clearance of damaged proteins associated with neurodegenerative diseases, the elimination of dysfunctional cellular components that contribute to cancer, and the metabolic adaptations that reduce the risk factors associated with cardiovascular disease and type 2 diabetes. And the Ayurvedic medical tradition, which prescribed specific fasting protocols for specific health conditions 2,000 years ago, was prescribing autophagy induction 2,000 years before the Nobel Committee recognized its discovery. Now, acupuncture. For most of the 20th century, the Western medical establishment's position on acupuncture was unambiguous. It was placebo. The effects reported by patients were the product of expectation and suggestion, rather than any biological mechanism produced by the insertion of needles into specific points on the body. And the Chinese medical tradition that had been mapping those points and documenting their clinical effects for approximately 2,000 years was classified as pre-scientific folk medicine, with no physiological basis. And then researchers began examining what actually happens in the body when acupuncture needles are inserted, and the findings produced by that examination have been sufficiently consistent and sufficiently replicable across independent laboratories that the position of the National Institutes of Health and the World Health Organization on acupuncture has quietly and significantly shifted from dismissal to conditional endorsement for specific conditions. And what the research has found is that acupuncture needle insertion at specific points produces measurable effects on the nervous system, including the release of endorphins and enkephalins, the activation of specific brain regions visible on functional MRI imaging, the modulation of the autonomic nervous system, affecting heart rate and blood pressure, and the stimulation of connective tissue responses that influence local and systemic inflammation. And a 2012 meta-analysis published in the Archives of Internal Medicine, analyzing data from approximately 18,000 patients across 29 high-quality randomized controlled trials, found that acupuncture produced significantly better outcomes than sham acupuncture and no acupuncture for chronic pain conditions, including back pain, neck pain, osteoarthritis, and headache. And the specific acupuncture points that the Chinese medical tradition identified 2,000 years ago as most effective for pain management correspond with a consistency that is not explained by coincidence to the locations of major nerve bundles and connective tissue planes whose anatomical significance modern neuroscience has only recently mapped. Now, meditation. The contemplative traditions of Buddhism, Hinduism, and Taoism have been prescribing systematic meditation practice for mental and physical health for approximately 2,500 years, with a specificity of claimed benefit that Western psychology dismissed as spiritual metaphor, until researchers began putting meditators in brain scanners, and the results produced by those scans have generated one of the most rapidly expanding fields in contemporary neuroscience. Because what the scans show is that regular meditation practice produces measurable structural changes in the brain. And the most significant finding, published by Sarah Lazar of Harvard Medical School in 2005, is that experienced meditators have measurably greater cortical thickness in regions of the brain associated with attention, interoception, and sensory processing than age-matched controls. Meaning meditation does not simply change how the brain functions in the moment of practice, but permanently alters its physical structure. And Jon Kabat-Zinn's mindfulness-based stress reduction program, which translated Buddhist meditation techniques into a secular clinical format, has now been tested in hundreds of randomized controlled trials, showing significant reductions in anxiety, depression, chronic pain, and the physiological markers of stress, including cortisol levels and inflammatory cytokines. And the Buddhist tradition that developed these techniques 2,500 years ago described their effects in language that modern neuroscience is now translating into the vocabulary of neuroplasticity and autonomic regulation. And the specific claim of the Buddhist tradition that the mind can be trained to produce specific physiological states through deliberate practice is the claim that the entire field of contemplative neuroscience has spent the last 20 years confirming. Now, the gut-brain connection. The Ayurvedic medical tradition has described the digestive system as the root of health and disease for approximately 3,000 years, using a concept called Agni, the digestive fire, to describe the relationship between the state of the digestive system and the overall physical and mental health of the individual, and the claim that mental and emotional states are directly produced by and responsive to the state of the digestive system was so far outside the Western biomedical model that it was not seriously investigated until the last 2 decades. And what the investigation has revealed is that the enteric nervous system, the network of approximately 500 million neurons lining the gastrointestinal tract, contains more neurons than the spinal cord, communicates bidirectionally with the brain through the vagus nerve, produces approximately 90% of the body's serotonin, and is populated by a microbiome of approximately 38 trillion microorganisms, whose composition has been shown to influence mood, cognition, immune function, and the risk of conditions ranging from depression to Parkinson's disease. And the field of psychobiotics, the study of how gut microbiome composition influences mental health, is one of the fastest growing areas in contemporary psychiatry. And a 2019 study published in Nature Microbiology, analyzing the gut microbiomes of over 1,000 individuals, found specific bacterial species whose presence or absence was significantly associated with depression and quality of life scores. And what the 3,000-year-old Ayurvedic tradition was describing when it said that digestive health is the foundation of all health was the gut-brain axis. And it was describing it not as a metaphor, but as a clinical observation derived from systematic attention to the relationship between what people ate, how their digestion functioned, and how they felt and thought and behaved. Now, willow bark. For thousands of years across cultures with no documented contact with each other, from ancient Egypt to ancient Greece to ancient China to indigenous North America, healers prescribed preparations made from the bark of the willow tree for fever and pain. And the Ebers Papyrus, dated to approximately 1550 BC, contains a description of willow bark preparation for pain relief. And Hippocrates, writing in approximately 400 BC, describes the use of willow leaf tea for fever and pain in childbirth. And traditional Chinese medicine incorporated willow bark into its pharmacopoeia for inflammatory conditions. And the active compound responsible for these effects was identified by European chemists in the 19th century as salicin, which the body metabolizes into salicylic acid. And the modification of salicylic acid into acetylsalicylic acid, which reduces its gastric side effects while preserving its therapeutic properties, produced aspirin, the most widely used drug in human history, and the specific therapeutic applications for which aspirin is now prescribed, pain relief, fever reduction, anti-inflammation, and cardiovascular protection through platelet aggregation inhibition, correspond precisely to the applications for which healers across every inhabited continent had been using willow bark preparations for thousands of years before the chemistry that explained those applications was understood. And the question is not simply that ancient healers were right about willow bark. It is that healers on every continent independently identified the same plant family for the same clinical indications without any knowledge of the biochemistry that connected the plant to the outcome, which implies a systematic process of clinical observation across generations that was producing pharmacologically valid results without the theoretical framework that modern medicine considers necessary for pharmacological validity. Now, cold water immersion. The practice of deliberate exposure to cold water for health benefits appears in the historical record of ancient Rome, ancient Greece, ancient Japan, and numerous indigenous traditions. The Roman thermae with their cold plunge pools, the Japanese practice of misogi purification in cold water, the Scandinavian tradition of cold water immersion following sauna, and the consistent dismissal of these practices by 20th century medicine was that they were cultural rituals with no biological basis beyond the subjective experience of invigoration that cold water produces. And then, researchers began measuring what actually happens in the body during cold water immersion, and the findings produced by that measurement have been sufficiently consistent to generate serious clinical interest. Because cold water immersion produces a significant release of norepinephrine in the brain, with studies showing increases of up to 300% following cold water exposure. And norepinephrine is a neurotransmitter and hormone involved in mood regulation, attention, focus, and the body's inflammatory response. And the specific mental health benefits claimed by practitioners of cold water immersion, improved mood, reduced anxiety, increased mental clarity and energy, map directly onto the documented neurochemical effects of norepinephrine release. And Wim Hof, whose extreme cold exposure practices initially attracted ridicule from the medical establishment, has now been the subject of peer-reviewed research, including a 2014 study published in the Proceedings of the National Academy of Sciences, showing that trained individuals using his techniques could voluntarily influence their immune response in ways that the existing immunological model said was impossible. And what connects all of these ancient practices across the specific differences of their cultural contexts and their geographic origins is not simply that they work, because the clinical evidence now confirms that they work, but that the people who developed them were doing something that deserves more credit than the word superstition allows. They were conducting systematic clinical observation across generations, tracking inputs and outcomes with sufficient consistency to identify real biological relationships between specific practices and specific health outcomes.